Welcome everyone to the next distinguished lecture of the Josep Carreras Leukaemia Institute. It's a great pleasure to have with us Professor Mark Dawson. He's a clinician scientist and associate director of research translation at the Peter MacCallum Cancer Centre in Australia. And he has been studying epigenetics from a basic standpoint and more translational in development and in cancer. And in this field, he has moved forward several first-in-class epigenetic therapies, and he's involved in many clinical trials with these drugs. He's also a professor at the University of Melbourne and a Sir Edward Dunlop Fellow of the Cancer Council of Victoria, and also Howard Hughes International Scholar. He has been recognized by different awards and memberships. For example, the Australian Academy of Science. He's also an EMBO member, and among the awards, just to mention the MacCallum Award from the International Society of Experimental Hematology, the Jacques Miller Medal from the Australian Academy of Science, and the Prime Minister Prize of Life Scientist of the Year in 2020, so just last year. So it's a real pleasure to hear the science from Mark. Remember to send your questions to the question mark button that you have there, and the floor is yours, Mark.
Manel, let me start by just thanking you again for the invitation. It's a real privilege and honor to be here, and I wish I was there in person, as we talked about before, but sadly today, at least, it'll be virtually. But hopefully, I will visit the institute and you in the next year. So thank you again for the opportunity to present our work. So today, I guess what I'm going to do is really start, and the major area I'd like to focus on is to really provide our perspective on balancing the contributions of both genetic and non-genetic mechanisms to malignant clonal fitness. In the second part of my talk, I'll talk to you a little bit about some of the chemical biology work that we've been doing in this area as well. Okay, so let's get started. Much of the way we think about cancer is really driven from what we have gleaned through bulk genome sequencing, and then the subsequent inference of variant allele frequencies that inform when the particular mutations arise. And this has really given rise to this imagery, which is very familiar to many of you, known as a fish plot. And what the implication here is that cancer arises and progresses through linear sequential evolution. And the time frame on which this occurs is relatively short. And this concept is actually, is quite pervasive. It really influences the way we think about many malignancies and how they develop. And here I'm going to be giving you the example of clonal hematopoiesis. We're learning more and more that many of our tissues have clonal populations, if not all of them, as we age. And this has probably been best studied in clonal hematopoiesis. And here, this thinking of linear sequential evolution really comes to the fore. So what we believe happens here is that a mutation arises, often in epigenetic regulators, conferring upon these cells a survival and proliferative advantage, and they become the dominant population. We then believe that this sets the stage for a new mutation to arise within the clonal hematopoietic clone that then gives rise to a myelodysplastic syndrome. And this then sets into motion the inexorable progression from myelodysplasia to acute myeloid leukemia. And again, here we believe that the mutation arises in the myelodysplastic syndrome clone, which originated from clonal hematopoietic clone. So this is the way we think about things. But when we start to analyze this at single cell resolution, much of this thinking has started to change. And here I'm showing you some excellent work from Amit Verma and Uli Steidel's lab, where they really looked at this linear sequential model from clonal hematopoiesis to AML at single cell resolution. Now, what they found was that actually much of our thinking needs to be rethought and challenged, because what they found was the cells that gave rise to myelodysplasia, the malignant stem cells that gave rise to myelodysplasia, were actually quite different to the malignant stem cells that gave rise to acute myeloid leukemia. And they really focused on the fact that much of this genetic diversity exists in the hematopoietic stem and progenitor cells, and potentially much earlier than we ever expected. And this really has been taken forward here, and I'm giving you two examples now of studies, one that is published and one that is still yet to be published from Jyoti Ngalia's group. And here, essentially, what these investigators were looking at, were trying to infer the timing of these mutations. And they looked at a relatively genetically simple disease, polycythemia vera. So we've known for nearly 16 years now that polycythemia vera almost inevitably contains a gain-of-function mutation in the non-receptor tyrosine kinase JAK2. And here is a patient that is diagnosed at age 65, which is when most patients are diagnosed, around in the 5th and 6th decade of life. This sample itself was taken at age 82, and using a method that I don't really have time to go into, they were able to take single colonies, sequence them, and work backwards to infer the timing of these mutations. Now, interestingly, even though this patient presented at age 65, it is likely that the seminal driver mutation for this disease originated as early as 1.8 weeks post-conception, and certainly within the first decade or so of life. So this really challenges this concept that linear sequential genetic evolution is rapidly driving the processes that enable clonal fitness in cancer. So now if we go back to this fish plot, this is what we infer from bulk genome sequencing, but potentially actually what we're really seeing is the fact that much of this genetic diversity occurs very early, maybe even as early as in your 20s. And what we're seeing is the dominance of populations most adept at dealing with the metabolic, hypoxic, and nutritional pressure, and including therapeutic pressure. Now, I want you to bear this diagram in mind because, to be honest, we don't really have any proof that this is indeed the case. And hopefully by the end of this first part of my talk, I will provide some of this proof. This pervasive thinking of linear sequential evolution also, it permeates into the way we think about therapeutic resistance. So acute myeloid leukemia, like many malignancies, has a hierarchical structure, and its apex contains the rare cancer stem cell, the leukemia stem cell. And although acute myeloid leukemia remains, an incurable disease for the majority of our patients, actually our therapies work relatively well. When we consider this to what we see in the context of therapies with solid malignancies, induction chemotherapy in acute myeloid leukemia actually puts up to 80% of our patients into a complete remission. But sadly, for most of them, that remission is short-lived. And we think that that is the case because we failed to eradicate the cancer stem cell and the disease comes back. What drives this resistant process? We often use DNA damaging agents as part of our treatment, the backbone of which hasn't changed since the 1970s. And we believe that this chemotherapy induces new mutations that confers upon the cancer stem cells a genetic clonal advantage, ultimately driving the relapse. And this is reinforced here again by more fish plots that suggest that this clone here, which arose a few months after the initiation of therapy, becomes then the dominant clone at relapse. But I want you to bear that in mind with what I've been telling you before about how quickly genetic evolution happens in the timescale of that. So we really started to challenge this paradigm initially when we were trying to model how resistance to an epigenetic therapy arose. And really here, I want you to take home two major take-home messages from this paper. They were the two major paradigms I hope we tried to establish here. Firstly, when we looked at the fully resistant population and compared it to the drug-naive population, there was actually no evidence of any genetic mutations. What we did, however, see is that resistant population was more immature in its phenotype, resembling that of leukemia stem cells. Not only did they look like leukemia stem cells, they had the transcriptional program of leukemia stem cells, and also we functionally validated in vivo that they were markedly enriched for leukemia stem cells, up to 100,000-fold enriched for leukemia stem cells compared to the drug-naive population. Now, this was an experimental therapy in an experimental model, and the clinical significance of this remained unclear until this paper a couple of years later that was published by Liran Slush and when he was working with John Dick. And here Liran, essentially what he did was look at the pre-treatment samples for patients with acute leukemia treated with conventional chemotherapy. Again, looked at them at the time of remission and again at relapse. And using both genomics and functional assays, he found that it happens is at the time of relapse, this population is markedly enriched for leukemia stem cell properties. And when he looks at the genomics, there was actually no evidence in any of the patients that they studied that there were any coding mutations that could account for the relapse phenotype, the relapse and resistance phenotype. In fact, when we've now gone back and looked at all the paired published samples by whole genome or whole exome sequencing, what we clearly see is that up to 40% of patients, their fish plots actually look like this, which is what you start out with, is what you end up with. You might get some clonal extinction, but there's no clear evidence of any new dominant driver mutations that are seen. And in fact, even in the 60% of patients where there's new mutations at the time of relapse, there's no functional evidence really to say that that is really what drove the relapse. And so this really made us, come to the concept that we need to start thinking about non-mechanisms of clonal fitness and adaptation. And if you buy into this, given the fact that there is overwhelming evidence for this, that intratumor heterogeneity is not always driven by mutations, what then are the non-genetic mechanisms that drive tumor initiating capacity? What drives clonal advantage and clonal evolution within various tissue microenvironments? What drives drug resistance in the absence of the development of new mutations? And how do malignancies start to evade immune surveillance? We realized we had to do this at single cell resolution and actually in a more sophisticated manner. And this was not uncommon because at the time that we started this work nearly 4.5 years ago, single cell technologies were well and truly taking off. The problem with single cell RNA sequencing is that it provides a really detailed but nonetheless snapshot in time. And you never really understand the transcriptome of this yellow cell enables it to decide to become a purple cell or a blue cell. Now, you can do this through DNA barcoding and lineage tracing. This is a destructive process that you don't capture the RNA. And we, like many others, started to couple the two. And the method we developed is a method called Splinter. Now, Splinter enables us to really capture an expressed barcode in up to 90% of the cells without any preamplification straight off the 10x machine. It is incredibly well correlated with DNA barcoding. So here is a population of cells where we have assessed clone size as evidenced by the size of the bubble and clone number as evidenced by the number of bubbles in different colors. And when we assess this, either by DNA barcoding or by single cell RNA sequencing, you can see that the results are absolutely cool. We also optimize this method to enable the fact that we always have, or most frequently have, a single barcode in a cell. And in the very rare circumstance that we get a doublet, we've developed computational methods to really track this as well. We didn't just stop with one barcode library, we built very many. I think we're up to 8. And really, here's an example of our first 3. They all have a 60 base pair barcode structure, and I can give you the reasons for why we chose that later on. And really, we developed many because we wanted to track multiple genetically distinct populations, as I'll show you through my talk, and assess their clonal behavior in vitro and in vivo. So the next thing I want to do is really focus on our method. And this is a really important slide to understand the remainder of this section of my talk. So what we do here is we work with primary malignancies, and in this case, I'm showing you our work with a murine model of acute leukemia. So we take these primary leukemia cells and we barcode them. And then we enable those cells to grow in culture for a short period of time, less than a week. But what this allows us to do is that every one of these barcoded clones is able to divide several times over, producing identical progeny. Those identical progeny enable us to keep some aside for various single cell methods, either single cell ATAC-seq or single cell RNA sequencing, and then the rest of them we transplant in vivo into mice, whereby each mouse gets an identical compendium of all the clones within the starting population. We then harvest these mice at a particular time point, be it a disease progression, be it a therapeutic resistance, as I'll show you soon, and then again assess this by single cell technologies. And this approach allows us to understand what cancer cell behaviors are inherent to the clones, what are acquired in vivo, and what those processes of adaptation are that enable clonal dominance. I told you we wanted to study non-genetic mechanisms of cancer cell fitness. And actually there are some rare but frequent examples of this. Right? And they are primarily pediatric cancers, where one mutation is really all it takes. In hematological malignancies, a really good example of that is those malignancies driven by MLL fusions. Now, for those of you who are clinically trained in the audience, in particular pediatric hematologists, you will know that if a child presents with an acute leukemia driven by, let's say, MLL-AF4 at age 8 and has no other mutations in their cancer genome, which is not infrequently the case, and a child presents with the same fusion, MLL-AF4, no other mutations, at age 8 months, invariably, the younger child, the 8-month-old, the infant leukemia, does worse. And that outcome cannot simply be explained by additional mutations in the cancer genome. So we wanted to really use this as the paradigm to study non-genetic mechanisms of adaptation. So one of the things that is really paramount in the field is the cell of origin that may drive these cancers. And it's really been shown by a number of people in the field, principally Scott Armstrong and others, that the cell of origin really matters in terms of cancer cell behavior. And so here, what we did was we used a hematopoietic stem cell and a committed progenitor, in this case, a granulocyte macrophage progenitor, as the cell of origin for the acute leukemia. And when we transplant these in vivo, what we see is that at an equal time point, the disease burden generated by cells that have a cell of origin from a hematopoietic stem cell versus a GMP is virtually identical. Interestingly, actually, when we mix these populations in a 50-50 mix and transplant them and enable this different cell of origins to compete in vivo, again, surprisingly, what we see is that the number of clones that give rise to the resultant leukemia is virtually identical, and the clone size is also virtually identical. But one of the striking things we saw whenever we did this assay is that in every single mouse that we transplanted, and I'm telling you, as I told you before, we transplanted identical clones, whether that clone was derived from a GMP or from an HSC, it was the same clones that gave rise to the leukemia in every single mouse, suggesting that there is this cell intrinsic program that drives clonal dominance. Now, I told you one mutation is all it takes, and that is certainly true, as evidenced here by clinical sequencing from MLL leukemia patients. Many of these, regardless of diffusion, don't have any co-mutations. But not infrequently, there's a subclonal architecture to MLL leukemias, and this frequently involves mutations in the tyrosine kinase FLT3 or in the GTPases N and KRAS. So to study these and to understand how cooperating mutations may affect clonal cell behavior, we made several different models. And here, the MLL fusion was expressed along with a knock-in of the KRAS mutation or FLT3, also the mutation expressed from the endogenous allele. So now let's see how these three different genotypes behave. So one has just the MLL fusion, the other has a FLT3 activating mutation, and the other a KRAS activating mutation. So I want to spend some time just explaining this result right here. So here we were assessing clonal dominance ex vivo. Which cells grow best ex vivo? Many of our assays are done ex vivo. And here you can see this clone here in green is the most dominant cell, is the most dominant clone ex vivo. But then we transplanted these into 4 separate mice, and then we assessed the tissue burden in the bone marrow in dark blue, or in the spleen and extramedullary tissue in the light blue in all 4 mice. What we see is absolutely remarkable, at least to me, which is that the clones that drive the leukemia in every single mouse are absolutely identical. Not only is clonal fitness and clonal dominance a cell intrinsic process, and you'll see here that tissue predilection is identical. In all cases, the clone sizes were virtually identical in every single mouse. And here, as evidenced by this green clone, it's always larger and thrives more in extramedullary tissue compared to the bone marrow. This clone here does the opposite. We want to understand whether this was just true for leukemias just driven by an MLL fusion, but actually when we look at those that have a cooperating mutation in KRAS or FLT3, we see identical properties. And this can be explained because the transcriptome of these cells, these dominant clones, they actually all cluster together. This is a polyclonal population with the shared transcriptional features, regardless of the genotype. So let's look into what some of those transcriptional features of dominance might be. Well, prior to doing that, let me just provide you with some evidence that this is all non-genetic. So, we sequenced all of these tumors, both prior to transplantation, after primary transplantation, and after second tertiary transplantation. And what we see is that, as is well known with MLL leukemias, this is a genetically stable disease. Less than, even after tertiary transplants, less than 0.5 somatic mutations per megabase is seen. There is no copy number changes and no clear evidence of mutations in any of the coding genes that may drive leukemia. We also went to the trouble of seeing whether expression of the fusion itself, the MLL fusion itself, might drive clonal dominance. And you can see here, although the fusion is expressed fairly homogeneously in the starting population, here are the dominant clones, there's no clear evidence that actually the fusion was overexpressed, giving rise to this clonal dominance. So what then are the major transcriptional features? Well, we saw that the leukemia stem cell program was inherently active in cells that went on to give rise to clonal dominance. However, it's really important, it was not a prerequisite. There were many clones that were very dominant that did not have the quintessential leukemia stem cell program on. What we did see was there is a decrease in antigen processing and presentation in all of the genotypes that were transplanted that gave rise to clonal dominance, suggesting that evading immune surveillance, as many of this was done in immunocompetent mice, was very important. So I told you about the leukemia stem cell program, which has increased, but then when we also look at what other genes might be increased, we came to notice that every time we did these assays, there was increased expression of this gene called SLPI. If you've not heard of it before, don't worry, I had not either 3 years ago. It's a secreted leukocyte peptidase inhibitor, and its role in acute leukemia has not at all been studied. So we and others have spent a lot of our time looking for genetic dependencies in acute myeloid leukemia using various genetic screening processes, RNAi, CRISPR, and so on. And we've never found SLPI. Right? And consistent with that, actually, if we knock out SLPI in vitro, there is no evidence of clonal disadvantage. However, now when we knock out SLPI in vivo and transplant either a mix of SLPI knockout and SLPI replete cells, and this is a polyclonal knockout, we very clearly see clonal disadvantage regardless of the mouse background and regardless of the tissue that we assess, either extramedullary tissue or bone marrow. In fact, when we do this with clean SLPI knockout clones, the result is even more impressive. So here in yellow is a SLPI knockout clone and a SLPI wild-type clone transplanted in a 50-50 mix. And you can see the disease latency not very different to SLPI wild-type cells. However, all of the cells that give rise to leukemia, as you can see in the flow plot here, are SLPI wild-type, and none of the SLPI replete cells, depleted cells, actually are able to establish clonal dominance in this mix. And in fact, if you look at the clone, when we transplant a clone of cells that are SLPI deficient, some of these mice never come down with disease. So this is a really good example of how Splinter has enabled us to uncover transcriptional targets and potentially therapeutic targets, as we've been working on with this, that could not have been uncovered in vitro. I now want to move to look at some of the other things we discovered with Splinter. We often think about cancer stem cells as giving rise to the ultimate tumor burden with equal output. Every cancer stem cell behaves identically, we often think, and therefore the ultimate tumor burden is composed of an equal representation of all of the potential cancer stem cell clones. Another way of thinking of this is actually that might not be true, and cancer stem cells, much like normal tissue stem cells, as has been shown really well by Vallejo and Fernando Camargo's group, actually might have disproportionate clonal output. So to look at this, we employed Splinter, and you can see here that we started off with clones that were present in the starting population prior to transplantation in equal amounts. But in every single mouse that we transplanted, the top clone was a high output clone. It gave rise to over 95% of the disease burden. This we call a mid output clone because it rapidly multiplied and became much greater in size. And this we call a low output clone in that it was just sufficient to renew itself. Now, I told you that none of this clonal behavior will be explained by the cancer. The genome is identical in all of these cells. So we wanted to understand how much of this clonal behavior is heritable, how much of it is truly epigenetic and remembered. So what we did there was we rebarcoded these cells with another Splinter library. The different Splinter libraries enable us to do this, and then transplanted these into both immunocompetent and immunodeficient mice and assessed clonal output in both the extramedullary tissues as well as in the bone marrow. And what we see is very clear. Regardless of whether we look at immunodeficient or immunocompetent mice, whether we look in the spleen or in the bone marrow, clonal behavior is remembered in serial transplantation. High output clones stay high output. Mid output clones never transition to being high output. They stay mid output. And low output clones never transition upwards either. They largely stay as low output. We thought actually this clonal behavior and this behavior of malignant stem cells might be very important in understanding therapeutic resistance. And to really to look at this, we treated acute leukemia mice with a regime that really parallels what we treat our patients with. We treat our patients around the world really with a combination of cytarabine and an anthracycline. We usually give them 7 days of cytarabine and 3 days of an anthracycline, and that's why the regime is called 7 and 3. We have to adapt that to the mice, so we gave the mice 5 days of cytarabine and 3 days of an anthracycline. We can't use daunorubicin or idarubicin in mice, so we had to use doxorubicin. So this is very close to what induction therapy is in patient samples. And here now we took samples just prior to the commencement of treatment, 2 days into treatment, at the completion of treatment, and 2 days after treatment to see what happens to minimal residual disease. Unsurprisingly, at these 4 different time points, we have very different transcriptional signatures. This in itself is not surprising. But then we started to see other things. Interestingly, at day 2, what we saw is that a number of cells that subsequently went on to survive, because we could do this single clone resolution, actually adopt this process of senescence. And when we were studying this, these 3 papers were published, and they really described a very similar phenomenon whereby cancer cells under therapeutic pressure adopt this state of animated suspension, which they term diapause. Diapause is really a developmental process, and senescence and diapause, I guess, are relatively similar at least in the transcriptional world, because the signatures that were described here are exactly the same as those that we see in our senescent-like cells. Interestingly, this is a transient process, and the cells quickly recover, as you can see, by day 5 and day 7. They're really emerging from this shutdown in both cellular metabolism and cell cycle. Now, because we were able to do this at single clone resolution, we could understand this in much greater detail. Here's one of the most surprising things we found. Firstly, we found that when we track clonal output and its response to therapy, The mid and high output clones either take the hit and stay mid and high output, but more frequently, actually, as a consequence of the therapy, they change their clonal behavior. They go from high output clones down to low output clones. Even more surprising is the fact that the low output clones, in the context of therapeutic challenge, also change their clonal behavior and subsequently transition to becoming high output clones. Now, when we look at this clonal behavior in the context of the UMAPs of the adaptation process, what we see here again is that it's only the high output clones that undergo this transient process of senescence. The low output clones never do this. They just transition straight away into a high output state. And this is really important because if you look at the way these clones contribute to the tumor burden, in the absence of treatment, low output clones make up about 15% or so of the disease. But following treatment, the low output clones become the dominant population. They really transition to being the major cause for therapeutic relapse. And when you look at this adaptive process, it's very clear that high and low output clones adapt through largely distinct measures. Sadly, I don't have time to go through this in detail today, but you can see here, as evidenced by this heat map, the adaptive process is very different depending on the clonal output of that cell, inherent and cannot be explained by the cancer genome. Now, regardless of the adaptive process, what we found, if you remember what I showed you to start with as to why we got interested, here again, what you see is the process of adaptation converges on the fact that these cells that ultimately survive and come through the therapy come through with an enhanced propensity for leukemia stem cell properties. So now if we go back to what I started with, and we now look at this again, and if I take your attention here to this brown cell here, which is the dominant population after therapy. If we looked at this by bulk sequencing, we would never have detected this brown clone. And we would have thought that the therapy itself induced, let's say the brown clone had a P53 mutation, induced a P53 mutation, which drove the disease. What I showed you though, is actually all of this heterogeneity is pre-existing. And this clone here with this particular genotype actually had no reason to be advantageous at that time. However, in the context of therapy, was able to adapt best, change its clonal output, and came back as the most dominant clone, but it did not change its genome. So hopefully today what I've shown you is that cancer-initiating clones have a primitive cell of origin, and they co-opt pre-existing transcriptional programs of both self-renewal and immune evasion by reduced antigen presentation. I showed you how we use Splinter to find Slippy as a new regulator of clonal dominance that we only see in vivo. I showed you that clonal dominance is an inherent property regardless of the cell of origin or the genotype of the cancer cells, and this determines clonal output and tissue predilection. I showed you that malignant clonal output from cancer stem cells is not equal, but is a cell-intrinsic inherent trait. And whilst high and low output cancer stem cells can survive chemotherapy, the adaptive response is very different. And the therapy itself can alter clonal behavior. Most of all, I hope I've brought into your consciousness the fact that malignant cell behavior just cannot simply be explained by the cancer genome. And therefore, for us to achieve our ambition of precision medicine, we need to study both cancer genomics and the non-genetic mechanisms of adaptation and clonal dominance if we're truly going to improve the way we treat our patients and improve the outcomes. The work I've shown you today is in press and hopefully will be published within the next month. So now I want to actually transition to an area that my lab has not really worked on before, which is targeting cell death. And this has really become very important in acute leukemias because, as many of you know, the BH3 mimetics, and in particular venetoclax, which selectively targets BCL2, has really changed the face of many hematological malignancies and has changed the natural history of a lot of them. And much of this work has really been done with a dear colleague and friend who's a wonderful medicinal chemist, Rafael Rodriguez, who works at the Institute Curie. And it follows on from his work looking initially at seleninomycin, which is a drug that was developed initially by Bob Weinberg and Eric Lander, and thought to work through ion channels, sodium ion channels. Raphael made iterations of sillinomycin, ultimately ending up with a drug that we call ionomycin, and clearly showed that ionomycin actually works very differently. It works through the sequestration of iron. And as cell death is becoming so important in acute leukemia, we decided to work together to study ionomycin in the context of acute myeloid leukemia. So the first thing to say is that regardless of the genotype that drives the acute leukemia, ionomycin is very, very effective, killing most of these cells which broadly represent the genetic diversity seen in AML at low nanomolar concentrations, as you can see here. Interestingly, however, when we came to look at how ionomycin actually induces cell death, we started to get very surprised. Because unlike venetoclax, which induces apoptosis and a potent activation of caspases, the cell death induced by ionomycin does not really potently activate caspases. And consistent with this, actually caspase inhibitors can rescue the cell death induced by venetoclax, but not by ionomycin. Now, there are multiple other programmed cell death pathways that have been really well studied, and so we started to look at all of them. And what I'll tell you in brief is that it is not necroptosis, it is not pyroptosis. Ferroptosis is a possibility, but largely it can't be canonical ferroptosis, because although ionomycin induces lipid peroxidation and has some features of ferroptosis, the inhibitors or the inducers of ferroptosis, such as RSL3, work really well, and ferristatin, which is an inhibitor of the classical ferroptotic pathway, rescues this completely, but actually there is no rescue of the ionomycin-induced cell death. So we then turned to chemical biology, and using some of the chemical biology techniques that we've used previously, we use click chemistry to try and localize where in the cell ionomycin goes. And in acute leukemia cells, where it accumulates is again in the lysozyme. And you can see very clearly within the lysozyme, it accumulates there and it sequesters ferrous iron. And this increase in ferrous iron is highly, highly reproducible. One possibility that we entertained was this might lead to lysosomal immobilization and in turn cause cell death that way. Although I'm not showing you the data here, we largely excluded that as well. So as we had not clearly found a known pathway for cell death, we decided to use unbiased functional genomics, which is probably the main platform used in my laboratory, to try and look at what are the mechanisms of cell death. And here we did this in multiple cell lines. And when we look at the cells, the rare cells that survive ionomycin, what we found was actually the majority of the hits related to glycolytic metabolism, carbohydrate metabolism, and the structural and functional integrity of the mitochondria, as evidenced here by many of the major hits that we see that primarily function in either mitochondrial homeostasis and or glycolysis. And I'm highlighting here two of our top hits, HK2, which is one of the first enzyme pathway, and PGP, which is a repair metabolite enzyme, also critically important for glycolysis and also the pentose phosphate pathway. Now, I'm not going to show you heat map after heat map of global metabolomics, and we did all of that. And what it clearly showed us was ionomycin really uncouples the citric acid cycle and increases pathways that generate ATP by alternative mechanisms in order for the cells to survive. But if we now look at the two major hits, because it really was very clear, even from our global metabolomics, that glycolytic flux and the OXPHOS pathway were the key central metabolic processes targeted by ionomycin. What we see here is that if we grow cells in increasing concentrations of glucose, as would be predicted by the CRISPR screen, actually we increase the toxicity of ionomycin. A pathway to drive by is to shunt glucose metabolism down the pentose phosphate pathway. In fact, if we inhibit the pentose phosphate pathway, we actually also increase the toxicity of ionomycin. And if we use drugs that change the metabolic profile of the cells to anaerobic metabolism, we can now start to alleviate some of the major toxicities of ionomycin. But we never really fully rescue it. So the metabolomics and the CRISPR screen really told us that we should focus on the mitochondria, but we were also interested in what the major transcriptional changes were after 6 hours of ionomycin. What you very clearly see here is within 6 hours, you get a marked decrease in synthetic enzymes involved in mitochondrial metabolism and a marked upregulation of all of the regulators of mitochondrial stress. Whilst I'm not a great fan of gene set enrichment analysis, what I'm really showing you here is a marked increase in this mitostress response pathway. How important is this targeting of the mitochondria and potentially the uncoupling of oxidative phosphorylation? Well, if we look at this by a seahorse assay assessing basal and maximal respiration, what you clearly see is that ionomycin really affects this very clearly early on, and it's sustained. And this is a result of the fact that mitochondrial iron is decreased. The reason that mitochondrial iron is decreased is that the lysosome and the mitochondria actually shuttle iron between each other. And the sequestration of the iron in the lysosome leads to a decrease in mitochondrial iron, and this then results in increased reactive oxygen species. So we thought this might be the mechanism of cell death. But actually, it turns out that whilst N-acetylcysteine and other antagonists of ROS are very good at rescuing other drugs that induce cell death via increased ROS, it does not rescue the effects of ionomycin. So this then made us move on to examine the mitochondria at the ultrastructural level. And it became clear to us that our CRISPR screen was only very good at knocking out single genes, but actually mitochondrial outer membrane permeabilization requires the coordinated activation of both BAX and BAC. So here we created isogenic cells that were either wild type or BAX-BAC knockout. And here you can see the mitochondrial, the ultrastructure of the mitochondrial is largely similar. What happens with venetoclax? Well, you get MOMP, you get mitochondrial destruction, and all of this is completely preserved in the context of a BAX-BAC knockout. Interestingly, with ionomycin, what we see is that the very dysmorphic mitochondria is also seen in the BAX-BAC double knockout. You see evidence of elongated, stretched mitochondria, mitochondrial perforation of both the outer and inner membranes, and mitophagy. But what is remarkable to us is that despite the fact that the mitochondria look so abnormal, the cells no longer die. So it really told us that mitochondrial outer membrane permeabilization was very clearly the mechanism of cell death induced by ionomycin. So let's go through this in a bit more detail. And here I'm looking at assessing cell death either by PI exclusion or by ATP production. And you can see that venetoclax, actually, if you use it on BAX-BAC double knockout, you abolish cell death, and you also preserve ATP production. But interestingly, when you do this with ionomycin, ATP production is still markedly reduced. This was an important result because it really told us that the metabolic effects of ionomycin preceded the cell death. Now, again, this is confirmed by the seahorse assay, which again shows you that with venetoclax in the BAX-BAC double knockout, respiration is absolutely intact, whereas with ionomycin, this is still markedly perturbed. Now remember, mitochondrial outer membrane permeabilization actually leads to the activation of caspases. And if a picture tells 100 words, hopefully this video will tell several more, because what I want you to see here is the induction of cell death. So BAX and BAC will be recruited in this reporter cell line, and when the cells die, they will go red. And you see that with venetoclax, this is really rapid, and the cell death is markedly prolonged in the presence of caspase inhibitors. In contrast, when you look at cell death induced by BAX-BAC activation with ionomycin, in the presence or absence of caspase inhibitors, actually makes no difference. The cell death is not at all really prevented by caspase inhibition, as I'd shown you earlier. So again, we wanted to just confirm using an orthogonal method the mitochondrial outer membrane permeabilization was actually happening. So here we use biochemistry, and very clearly you see that when BAX relocates from the cytoplasm up to the mitochondrial membrane, there is a release of the proteins within the intermitochondrial space, including cytochrome C, which is one of the canonical ways in which we follow mitochondrial outer membrane permeabilization. So at this point, we realized that BAX-BAC activation and MOMP mediated ionomycin-induced cell death. But how was it activating BAX and BAC? Principally, we've understood this by looking at the BCL2 family of proteins, including the BH3 only proteins. And there are 8 that are the BH3 family proteins, and in our cells, actually only 4 of these are really well expressed. So what we did initially was make isogenic cell lines that knocked out each of these either in isolation or in combination. And what we clearly see is, as Yannis's group had previously shown, that either a single or a double knockout of Noxa or Noxa and Puma actually confers resistance to venetoclax. But in fact, knockout of all 4 of these really did not change the mechanism of cell death induced by ionomycin. The ionomycin was still able to activate BAX and BAC despite lacking Noxa, BIM, BID, and Puma in various combinations. What about overexpressing the BCL2 family of proteins? Here again, if you overexpress BCL2, venetoclax resistance occurs. But again, note that ionomycin activates BAX and BAC through this mechanism. To be fair, we don't really know how ionomycin activates BAX and BAC. And we've looked very hard for at least a year now. And it's possible that it might do so through membrane lipids, but that is still ongoing work. But importantly, what this enabled us to do was say, well, venetoclax and ionomycin both converge on mitochondrial outer membrane permeabilization. They do so by very different methods. They do also affect very different metabolic pathways. Would they work together? And the clear answer is there is marked synergy when these 2 drugs are used. As you can see, even with a really low dose of ionomycin, even 1 nanomolar of venetoclax is able to change the mitochondrial membrane potential and result in MOMP and activate cleave caspases. So what then in vivo? So here I'm summarizing some results and showing you that although ionomycin in low dose or venetoclax in low dose are unable to prolong the survival of the mice treated with this aggressive leukemia model, actually the combination, despite the fact that treatment was only for about 30 days, markedly prolongs the survival, and some of these mice never come down with leukemia. Now, venetoclax has changed the natural history of AML, and it's either given, to unfit patients with azacitidine or fit patients with chemotherapy. And here is a, transformative clinical trial combining chemotherapy with venetoclax. What you see is markedly improves outcomes for various genotypes of acute myeloid leukemia, but not so for P53, which remains the Achilles heel of too many therapies. And, consistent with this, we see marked venetoclax resistance with P53 knockout cells. But again here, we see absolutely no evidence that a P53 knockout AML would not be able to respond to ionomycin. And this really spurred us on to look at many of these patients who had actually relapsed and were resistant to venetoclax. And again here in primary patient samples, we show that the combination of ionomycin and venetoclax overcomes the therapeutic resistance. So I want to stop here and summarize this second and final portion of my talk. And this work was done by Sylvain Garcia, who's a wonderful physician scientist based out of France, who spent some time in my lab. So what Sylvain showed is that ionomycin induces cell death in a manner that is quite distinct to the canonical programmed cell death pathways. It results in mitochondrial stress, and it is particularly effective in cancers with a high glycolytic flux that depend on oxidative phosphorylation for generation of ATP. The effects of mitochondrial metabolism actually precede the induction of mitochondrial outer membrane permeabilization, and the MOMP induced by ionomycin, in contrast to the BH3 mimetics, does not actually result in potent caspase activation. BAX activation is clearly not dependent on the BH3 only proteins, nor is it alleviated by BCL2 expression. And as I said to you, we still don't know exactly how BAX and BAC are activated. I showed you that inomycin shows marked synergy with the venetoclax, and inomycin activity is also also enables us to overcome venetoclax resistance, including in p53 mutant cancer cells. I should also say that this body of work was also just this week accepted for publication, and hopefully will be out again in the near future. So I have the joy of working with a number of fantastic scientists. I work very closely with my wife, Sarah Jane Dawson, who runs an independent lab focused primarily on cancer genomics and the solid malignancies. But we share a lot of staff and students together. She looks at the world of cancer largely through a genomic lens, and me, as you've seen today, largely through a transcriptional and non-genetic lens. And hopefully together we see it a little bit more clearly. There are a number of people around the world to really thank for much of the work I presented today. This is where I work, which is the Peter MacCallum Cancer Centre. It's the only comprehensive cancer centre in Australia. And we treat our patients on these floors, and we do our research on those. And in that way, actually, there's a lot to be thankful for. And these are also the funding agencies to whom I'm very grateful for investing in us in trying to drive some of this science forward. So I'm going to stop there, and if we have time, take questions.
Thank you very much, Mark, for this wonderful lecture. I think that has arisen many questions and provided some answers also. So trying to organize here the questions that people has. One really directs to this particular protein that you have identified, sleepy.
Yeah.
So how is the expression in patients, in AML patients, and how is upregulated?
Yeah. So they're both really great questions, and I didn't have time to go through it today. It's all in the manuscript, but I'll tell you what we found. We found that SLIPI is regulated by the CEBP family of transcription factors, and in particular CEBP epsilon, which hetero and homodimerizes with alpha and beta. That is the principal mechanism by which SLIPI is regulated in myelomonocytic cells. SLIPI is secreted, and we think that much of its activity results in preparing the in vivo niche for the cancer cells. Right? And that is where its principal activity is. And much of our work now is looking at how we might therapeutically target this process.
Thank you. Another question relates, you have mentioned several times this non-genetic mechanism driving selection, clone evolution. So you go in detail, which are these non-genetic mechanisms? Which type?
Yeah, look, I guess, so there's a number of different things that we've described in the work. Many of these processes might relate to a certain transcriptional program. And I gave you one example of SLIPI, there's others in there as well. The chromatin landscape is something that we are continuing to study, how the chromatin landscape and the subsequent activity of transcription factors might mediate some of this clonal advantage. And this is, an area of work, given the fact that we're going to have to do this at single clone resolution. We're actually going to have to develop even more sophisticated technologies to the ones we've done to be able to monitor this serially and over time. One of the things that we noticed is that many of the clones that survive actually have transcriptional heterogeneity, and they have variations in transcriptional burst size and transcriptional burst frequencies, and many of the genes that have these variations that confer clonal dominance are genes that are known to be important in the pathogenesis of acute leukemia. Again, that is still a correlative finding, right? We still don't understand what drives the changes in transcriptional kinetics that underpin that process. How is that remembered and transmitted over multiple generations? This actually, I think this is the start rather than the end of what we're finding. What this work was mainly there to do was to raise the awareness that actually all cancer cell behavior just can't be explained by sequencing more cancer genomes. We have to think of other things as well that might be driving these features of clonal adaptation and clonal dominance.
And do you think that this mechanism of non-genetic adaptation that you have shown very relevant in leukemia, others in brain tumors in kids, are also relevant for the other solid tumors like lung, colorectal, or do you think that they have more mutations there? Or what do you think about this?
Yeah, I can tell you right now that we have been looking at many solid malignancies, breast, lung, others. These processes of cell intrinsic behavior is not just peculiar to acute leukemia. We see the same things in the solid malignancies that have, vastly different cancer genomes. They're, hundreds, thousands of mutations. But really what we're seeing is that single clone resolution, the clonal behavior is pre-programmed and it's identical regardless of how many mice we studied this
in. Yeah. Another question here. You show how HSCs and GMPs produce the same type of leukemia clones. Have you tested their behavior in the treatment conditions?
Yeah, excellent question. Really excellent question. And the short answer is no. Right? So what I showed you was all in the context of just establishing the leukemia. But what we now need to really go and do in follow-up work is look at whether the cell of origin, and I really want to emphasize that this is in this model of disease. I don't want people to walk away thinking the cell of origin doesn't matter. It clearly does matter, right? And we were just showing you here that regardless of the cell of origin, that cell intrinsic behavior is preserved, and that regardless of the cell of origin, these clones can still compete. So if you consider the fact that many cancers originate as a field defect in acute leukemia, we don't know what that is most of the time, except in the context of secondary leukemias driven by chemotherapy. It suggests that multiple cells of origin might be affected by the same initiating oncogenic event. And what I wanted the work to highlight was that actually, if the cell of origin is a progenitor or a stem cell, those progenitor cells can also compete and they can also contribute to the disease.
Yeah. Thank you. Another more technical, how you define dominant and minor clones in the chemotherapy treatment? How you focus only on down-regulated genes or only in those show different patterns of expression? How you do that classification?
Yeah, good. Because we were able to quantify exactly how much each clone contributed to the overall tissue burden, the overall tumor burden. We essentially nominated 2% of the disease burden or less as being a low output clone, and higher than 2% as being a mid or a high output clone, right? So if a single clone gave rise to more than 2% of the overall tumor burden, we would call it a mid or a high output clone. If it was less than 2%, we call it a low output clone. So that's how we classified those.
Okay. One or two questions more, and we'll let you go because we promised to close in 1 hour, more or less. So, did you evaluate some senolytic treatments here, combined with the standard chemotherapy, as an approach to avoid chemoresistance?
Yeah, it's a good question. So we didn't do it, but also, I guess, I use the word senescence and others have used the word diapause, neither of which I think are fit for purpose, because, this was such a transient process. It lasted, just until the completion of treatment, so days before the clones, adapt and rejuvenate themselves and are able to re-enter the cell cycle, turn back on their metabolic processes, etc. So I don't know how we would deliver a senolytic within that period of time. One of the things we're really interested in identifying is, can we prospectively identify cells that are likely to behave as low output clones, and what are the features of those, and can we design treatments that target those in combination. Because, you know, what I showed you is our conventional chemotherapies do pretty well at eradicating the high output clones. It's the low output clones that come back, right? You change the disease burden completely. You flip it on its head. Without any therapeutic challenge, 15% of your disease burden is by low output clones. After therapeutic challenge, 85% of your disease is low output clones, right? So they're the ones giving rise to MRD. They're the ones driving the relapse. So we need to find ways of prospectively identifying them and trying to eradicate them.
I guess the final questions in the interest of time. So you mentioned that you study different ways that adenomycin kills the cells. And did you study mitophagy?
Yeah, yes, we did. And we studied autophagy and mitophagy. And there are clearly features of that. You know, I showed you that there are ultrastructural features of these processes, but are they the main driving reason behind the cell death? The answer is no, because very clearly the only thing that completely rescues it is the prevention of mitochondrial outer membrane permeabilization.
Okay. Thank you very much, Mark. So I would like to remind that in 1 hour we have another lecture by Professor Yanis Aifantis here in 1 hour using the same connection. And I would like to take this opportunity to thank really Professor Dawson for his time and for this wonderful lecture. So thank you very much. Please join me.
Thank you, Manel. Thank you again.
Until next time. Take care.
Okay. Thank you.
Bye.
Bye.